In the quiet rhythm of life, beneath the surface of every living thing there lies a story, not written in ink or spoken aloud, but encoded in the fabric of our cells, a silent, unbroken script passed gently from generation to generation. This is the story of DNA and genes. As you rest here, letting your breath deepen and your thoughts soften, we will begin a slow, calm exploration of this delicate language of life. Not hurried or technical, but gentle and patient, like turning the pages of an old, treasured book under the light of a soft candle glow. Genetics, in essence, is the study of heredity, how traits are passed from parents to offspring. But more than that, it is a window into the instructions that quietly shape the form and function of all living things. From the smallest moss clinging to a shaded stone, to the wingspan of a bird gliding across morning skies, and to you, here, lying still in the hush of evening, all are guided by invisible threads woven within their cells. At the center of this story is DNA deoxyribonucleic acid. Though its name may seem complex, its purpose is elegantly simple. DNA is the molecule that carries the instructions for life. Every cell in your body, from your beating heart to the tiny hair follicle on your arm, contains this code. Quietly nestled within the cell's nucleus, DNA orchestrates the countless processes that allow a living body to grow, to repair, to sense, and to change. But this orchestration is not loud or chaotic. It is more like a soft symphony, countless notes played at once, blending into the background of life, guiding it gently, persistently. You cannot hear it, but it is always playing. As we begin this exploration, imagine shrinking down to the microscopic scale, floating slowly through the still, translucent interior of a cell. Around you, there is no harsh light, only a gentle ambient glow. You pass by jelly-like structures suspended in a quiet dance. There, at the very center, is the nucleus, a rounded sanctuary where DNA lives, coiled with care into tight spirals, waiting patiently to be read. Inside that nucleus, DNA is arranged in long, twisting strands that resemble a rope ladder, curled into a graceful spiral. This structure, known as the double helix, is both elegant and efficient. It holds the instructions needed to build every protein your body will ever require, and does so with astonishing precision. Each molecule of DNA is made up of smaller units called nucleotides. These are like the letters of a biological alphabet. There are only 4 adenine, thymine, cytosine, and guanine, abbreviated as A, T, C, and G. And yet, with just these 4 letters, the DNA in your cells spells out everything from the shape of your nose to the way your blood clots when you are wounded. These letters pair in a consistent and complementary way, A with T and C with G, forming the rungs of the spiral ladder. The sequence in which these letters are arranged becomes a kind of code, directing the synthesis of proteins and the regulation of processes. It is a language that the cell understands completely, though it is written in silence. Your entire genetic code, all of your DNA, is known as your genome. Remarkably, if all the DNA in a single human cell were stretched out, it would measure nearly 2 meters in length. And yet, it fits effortlessly inside a nucleus only a few micrometers wide. It does this by wrapping itself around small protein spools called histones, forming a dense, tidy package known as chromatin. This delicate folding is part of the elegance of the system, conserving space while still allowing access to the information when needed. You began as a single cell, a fertilized egg containing a unique combination of DNA from your mother and father. From that one cell, your body has grown to include trillions. And in nearly every one, the DNA remains the same, a blueprint duplicated with meticulous care, ensuring that your identity, your biological essence, is preserved throughout your life. Genes are the functional units within this long strand of DNA. Think of them as chapters within the book, each one offering instructions to build a particular protein or regulate a specific task. Some genes influence eye color, others help your liver break down toxins, and still others play roles in emotion, sleep, and memory. Most cells contain around 20, 0 to 25, 0 genes, each with its own quiet purpose. But not all genes are active at all times. Cells turn genes on or off in response to signals from their environment, a process called gene regulation. Like dimming a light, the cell can dial the activity of a gene up or down, adjusting its inner workings in response to subtle changes. This regulation is part of what makes life so adaptable, a gentle, responsive system constantly tuning itself to stay in balance. As we drift further into this journey, we will explore how DNA is copied, how genes are expressed, and how tiny variations in these sequences can lead to the wondrous diversity of life we see on Earth. But for now, let these thoughts settle in like the gentle fall of snow. Each breath you take is sustained by proteins your genes help to build. Each heartbeat is carried out by instructions long encoded in your cells. You are the living result of this quiet choreography of a code written in ancient molecules, spoken in no voice, yet understood by every part of you. In the chapters ahead, we will unfold this story one layer at a time, softly, patiently, without hurry. For now, as your body grows heavier and your thoughts begin to drift, know that within you lies a library, an archive of life's memory, resting in every cell, whispering in every heartbeat, guiding the rhythm of being, night after night. Part 2. What is DNA? Let us now turn our attention to a single, elegant molecule, one that quietly shapes every living thing you've ever seen, touched, or been. Deoxyribonucleic acid, more simply known as DNA. To describe DNA is to describe the quiet architecture of life itself. It is not a structure we see with our eyes, yet it defines what we are. It is not something we feel, yet it is woven into the essence of every heartbeat, every breath, every soft movement of the hand. DNA is a long, spiraling molecule that serves as nature's instruction manual. It tells each cell what it is, what it should do, and how it should grow. The DNA in a tree tells it how tall to grow, what shape its leaves should take, and how to repair itself if injured. The DNA in a hummingbird guides the rapid beat of its wings, the color of its feathers, and even the structure of its tiny heart. And the DNA in you, the same kind of molecule, built with the same basic components, quietly defines your traits, your cellular makeup, and the rhythm of your body's hidden inner world. The structure of DNA is often described as a double helix. Imagine a winding staircase, but not made of wood or stone. Instead, picture something soft and luminous, gently spiraling like smoke caught in still air. Two long strands wind around one another, like intertwining ribbons. These strands are made of repeating units called nucleotides, the small letters of life's alphabet. Each nucleotide consists of 3 components: a sugar, a phosphate group, and a nitrogen containing base. The sugar and phosphate form the sides of the ladder, the railings of the spiral staircase, and the bases, those nitrogen-rich components, reach inward to form the rungs, always paired in a very specific way. Adenine pairs with thymine, cytosine pairs with guanine, A with T, C with G. These base pairs are held together with delicate hydrogen bonds, not as strong as steel, but just strong enough. They open when needed, when the cell must read its instructions or make a copy. Then, like closing a well-worn book, they gently fold back into place. The precise order of these bases, the sequence, determines the information carried by the DNA. In this language, there are no spaces or punctuation, yet the cell can read it flawlessly, line by line, section by section, guiding every action and reaction within. There are about 3 billion base pairs in the human genome. That's 3 billion letters, silently ordered within nearly every cell of your body. And yet, despite the immensity of this code, over 99% of it is the same in all humans. We are, in our essence, remarkably alike. The small differences in sequence between us, just a fraction of 1%, help explain the diversity of traits, the subtle variations that make each person unique. DNA is often called a blueprint, but in truth, it is more than that. A blueprint is fixed and lifeless, an inert drawing on paper. DNA is active, dynamic. It responds to the environment. It folds and unfolds. It allows certain sections to be read more often than others, depending on what the cell needs. It is a living record, not in the way we usually use the word living, but in the sense that it participates, continually, in the process of life. Every time a cell divides, it must make an exact copy of its DNA. This process, known as replication, is carried out with astonishing care. The two strands of the DNA helix unwind, like the slow unraveling of a spool. Then, using each strand as a template, the cell builds two new complementary strands, creating two identical helices, where there was once just one. This ensures that when a cell splits in two, each new cell receives a faithful copy of the instructions. This process happens billions of times throughout your life, often without error. So precise is the cell's machinery that it detects and corrects most mistakes almost instantly. On occasion, however, small errors called mutations slip through. Some are harmless. Others can change a protein's structure or function. These changes are part of how evolution unfolds, the slow, quiet adaptation of life across time. But for now, as you drift further into calm, it's enough simply to imagine the DNA molecule for what it is, a graceful, silent scroll resting in the heart of your cells. It never stops working. It is copied, read, folded, and passed down. Every tree, every bird, every human carries this molecular whisper, this ancient song, echoing softly from one generation to the next. It is comforting to realize that the DNA in your body was inherited from countless ancestors, from people whose names you may never know, whose faces have long faded into time, but whose essence lives on in you. Their cells divided, their DNA copied, and in a long, unbroken chain, the message made its way to you. Not loud or boastful, just persistent, just quiet, just true. So as you rest now, with your thoughts dissolving into the background, let your mind picture the double helix, a pair of glowing ribbons, spiraling gently, reaching across time. Within that structure lies the map of life, one that speaks not in words, but in patterns, pairings, and sequences. A soft, unending language, patiently telling each cell what to do, when to grow, how to mend, and how to become part of something greater. In the next part of our journey, we will follow this molecule through time, through its discovery. We will look back to the early scientists who first glimpsed this hidden code, and slowly, step by step, piece together the shape of life's most essential molecule. But for now, let the thought of DNA, quiet, spiraling, ancient, carry you deeper into rest. Part 3. The history of DNA discovery. Let us now step softly into the past, into the quiet corridors of early science, where curiosity stirred in candlelit rooms and notebooks were filled by hand, page by careful page. The story of DNA is not just a tale of molecules and microscopes. It is also a human story of wonder, of patience, and of long hours spent peering into the hidden architecture of life. Though DNA was not named until the 20th century, its influence has always been part of life itself. The knowledge that traits pass from parents to children is ancient, observed quietly by farmers and herders who noticed that tall plants tended to produce tall offspring, or that certain sheep bore lambs with the same woolly curls. Long before anyone saw a gene, they saw its effects, subtle echoes of ancestry moving gently from one generation to the next. Our journey begins in the 1800s, in the monastery gardens of what is now the Czech Republic. There, a soft-spoken Augustinian monk named Gregor Mendel planted rows of pea plants, and watched them grow. His experiments were simple in appearance, crossing plants with different traits and counting the results, but behind the simplicity was deep insight. Mendel observed how certain traits, such as flower color or seed shape, reappeared in predictable patterns. He realized that some traits could be hidden in one generation and reappear in the next. He described this in terms of dominant and recessive factors, what we now understand as genes. Though Mendel had no concept of DNA, his careful observations formed the quiet foundation of modern genetics. Decades passed before the scientific world fully appreciated Mendel's work. Meanwhile, other researchers turned their attention to the cell itself, that tiny chamber of life where all activity begins. In 1869, a Swiss physician named Friedrich Miescher was studying the white blood cells found in discarded surgical bandages. In his lab, he isolated a strange jelly-like substance from the cell nuclei. It contained phosphorus, unlike most proteins, and it resisted the usual stains and solvents. He named it nuclein, a term that would eventually evolve into what we now call DNA. At the time, no one suspected this substance carried the code of life. Proteins were considered the most likely candidates for hereditary material, given their complexity and diversity. DNA, in contrast, seemed too simple, just 4 chemical bases, repeated over and over. It would take another half-century for the tide of understanding to turn. In the 1920s, British bacteriologist Frederick Griffith made an unexpected discovery while studying pneumonia in mice. He worked with 2 strains of bacteria, one deadly, one harmless. When he killed the deadly strain with heat and mixed it with the harmless one, the mixture still caused disease. Some transforming principle, had passed from the dead cells to the living ones, giving them new instructions. The identity of this mysterious factor remained unknown, but the idea that information could be transferred from one cell to another like a whispered secret was now firmly on the scientific stage. Then, in 1944, a quiet but pivotal shift occurred. Oswald Avery, along with his colleagues Colin MacLeod and Macklin McCarty, repeated Griffith's experiment and carefully purified the transforming substance. Their conclusion: the material that carried genetic information was not protein, as many believed, but DNA. It was a remarkable claim, met with hesitation by some in the scientific community. DNA was still seen by many as too repetitive, too uniform, to encode something as complex as life. But the evidence grew steadily, and minds began to change. The structure of DNA, the way its parts fit together, remained a mystery. That changed in the early 1950s, with the convergence of several scientific paths. Rosalind Franklin, a brilliant X-ray crystallographer working in London, captured stunning images of DNA using a technique called X-ray diffraction. One particular image, known as photograph 51, showed a clear cross-shaped pattern, the shadow of a spiral. It hinted at a helical structure, a turning ladder of molecules. At the same time, 2 scientists, James Watson and Francis Crick, were building models of DNA at the Cavendish Laboratory in Cambridge. Guided by Franklin's photograph and the chemical insights of Maurice Wilkins and others, they assembled cardboard and metal pieces into different shapes, trying to uncover the molecule's hidden geometry. Finally, in 1953, Watson and Crick proposed a model that fit the clues perfectly: the double helix. 2 strands wound around one another, with the bases A, T, C, and G paired like rungs on a spiral staircase. The structure not only explained how DNA could store information, but also how it could be copied with precision. It was a moment of profound clarity, drawn from years of steady, quiet effort by many minds. Their findings were published in Nature, alongside contributions by Franklin and Wilkins. The double helix was elegant, simple, and deeply powerful. It became an icon not just of biology, but of the beauty that science can reveal when we look deeply and patiently. In the years that followed, the understanding of DNA advanced rapidly. Scientists learned how DNA is read and translated into proteins. They mapped genes, unraveled the full sequence of the human genome, and began to explore the ways in which genes are turned on and off, edited and regulated. But the core idea remains the same, that in every cell of your body, in every seedling, every sea creature, and every blade of grass, there is a quiet scroll of DNA, containing instructions shaped over billions of years. The journey to understand this molecule has been long and collaborative, filled with quiet breakthroughs and gentle revelations. It is a story that reminds us that the deepest truths are often found not in the loudest places, but in the stillness of thought, in the patient unfolding of a spiral too small for the eye to see, yet vast in its importance. As you settle now, deeper into rest, imagine those early scientists not racing for fame, but gently following the evidence, step by step, like footprints in fresh snow. They brought into focus something ancient, something soft, something enduring. In the next chapter of our journey, we will explore the components of DNA more closely, the nucleotides, the sugars, the bonds. But for now, let the spiral shape of the double helix rest softly in your mind, turning slowly, endlessly, like a lullaby written in molecules. Part 4. The components of DNA. Now that we've gently traced the path through history that brought DNA into focus, let us turn our attention to the molecule itself, not in a hurried way, but with the same soft curiosity one might bring to observing a snowflake or a seashell. DNA, though small beyond measure, is a marvel of structure and symmetry. Its beauty lies in its simplicity, a handful of elements arranged with extraordinary care, repeating in elegant patterns that sustain all living things. To begin, let us return to the image of the double helix, that delicate spiral staircase winding through the quiet center of the cell. It is not one solid piece, but rather a structure composed of many smaller building blocks. Each of these blocks is called a nucleotide. Like tiles in a mosaic or notes in a lullaby, nucleotides are simple on their own, but powerful when arranged together. Each nucleotide has 3 parts: a sugar, a phosphate group, and a nitrogenous base. The sugar, known as deoxyribose, gives DNA its name, deoxyribonucleic acid. It is a 5-carbon sugar shaped like a gentle ring, its atoms joined in quiet coordination. This sugar forms the central scaffold of each nucleotide, like the trunk of a branching tree. Attached to the sugar is a phosphate group, a small cluster of phosphorus and oxygen atoms. Though tiny, it plays an essential role. It links one nucleotide to the next, forming the sturdy backbone of the DNA strand. Together, the sugars and phosphates form the sides of the ladder, running in opposite directions like mirrored rails. This arrangement gives DNA its polarity, with each strand having a beginning and an end, one side called the 5' end and the other, the 3' end. These numbers refer to specific carbon atoms in the sugar ring, a quiet numbering system that helps scientists navigate the twists and turns of the molecule. The third part of the nucleotide, and perhaps the most celebrated, is the nitrogenous base. These bases extend inward from the sugar-like steps, reaching toward the center of the helix. There are 4 types: adenine, A, thymine, T, guanine, G, and cytosine, C. Each is shaped just right to pair with another, forming the rungs of the DNA ladder. Adenine always pairs with thymine, and guanine always pairs with cytosine. These pairings are not arbitrary. They are held together by hydrogen bonds, gentle forces that are strong enough to keep the strands together, but light enough to be broken when the cell needs to read or copy the code. A and T form 2 hydrogen bonds, while G and C form 3, giving each pairing a distinct stability and spacing. This specificity, A with T and G with C, ensures that the genetic message is passed on with remarkable fidelity. The bases themselves are divided into two categories. Adenine and guanine are purines slightly larger, with a two-ring structure. Cytosine and thymine are pyrimidines smaller, with a single ring. This pairing of one purine with one pyrimidine keeps the width of the DNA strand consistent, like perfectly matched teeth in a zipper. When the DNA molecule forms its double helix, the two strands twist gently around one another, with the bases paired in the center and the sugar phosphate backbones wrapping along the outside. Each full turn of the helix spans about 10 base pairs, a softly repeating rhythm, like the slow turn of a windmill in a breeze. But the arrangement of these components is more than just structural. It is informational. The order of the bases, the sequence of A, T, C, and G, forms the genetic code. Every gene, every trait, every instruction begins with a particular sequence. In this way, DNA is not unlike a language, where letters form words and words form sentences. Instead of 26 letters, it uses only 4, and instead of forming stories, it forms proteins, the working molecules that carry out nearly every function in the body. Proteins are made of amino acids, and the DNA code tells the cell which amino acids to string together, and in what order. Each group of 3 bases called a codon specifies one amino acid. For example, the codon ATG signals the amino acid methionine, which often serves as a gentle starting point for protein synthesis. Other codons signal different amino acids or instruct the cell to stop building. In this way, a long string of bases can be read like sheet music, guiding the cell's machinery to produce the precise proteins it needs. These components, sugar, phosphate, and base, repeat endlessly, forming the long strands of DNA that coil and twist inside the cell. And yet, within that repetition lies infinite possibility. The same 4 bases are used by every living organism on Earth, from the simplest bacterium to the tallest tree to the human mind. It is a shared alphabet, a common thread that connects all of life. It is humbling to realize that your DNA, the precise sequence of bases that makes you who you are, was shaped not just by your parents, but by generations before them. Small variations in the sequence, called polymorphisms, accumulate over time. Some of these changes are passed on, some are not. This gentle flow of genetic variation, guided by inheritance and shaped by chance, is what gives rise to the quiet differences among us. The curve of a nose, the color of an iris, the sound of a voice. And yet, for all our uniqueness, the structure of DNA remains the same, a silent helix, composed of repeating nucleotides, forming the basis of life in every corner of the natural world. As you lie here now, calm and still, picture this spiral structure, soft and glistening, suspended like dew in morning light. Picture the bases pairing gently, the sugar phosphate backbone winding alongside, and the whole molecule resting quietly in the heart of the cell. You carry this structure in every part of you. It has been with you from your very beginning. It will be with you for as long as you live. In every moment, it offers its quiet instruction, not loudly, not urgently, but with gentle constancy, like a lullaby that never ends. In the next part, we will explore this spiral ladder in more depth, looking at how the double helix folds and functions. But for now, let your thoughts drift like mist over a meadow, carried gently by the image of DNA, intricate, ancient, and endlessly unfolding. Part 5. The double helix in detail. As we continue our gentle descent into the quiet world of DNA, let us now slow our focus even further to the graceful structure itself, the double helix, a symbol of life's continuity. A design as elegant as it is enduring. You might imagine the double helix as 2 slender ribbons spiraling around each other, like ivy coiling up the trunk of an ancient tree. But this spiral is not just beautiful, it is deeply functional. Every twist, every bond, every turn of the helix serves a purpose, shaped by billions of years of quiet refinement. Let us begin with the shape. The two strands of DNA curve around one another in a gentle, right-handed spiral. This right-handedness means that the helix winds in the same direction, that the fingers of your right hand curl if your thumb is pointing up, a subtle, structural choice written into nature's blueprint. The spiral repeats every 10 base pairs, forming one complete turn every 3.4 nanometers. These dimensions may be minute, far beyond the reach of the naked eye, but they give DNA its precise geometry, a steady, predictable pattern, like a soft rhythm, repeating in time. Each of the two strands of DNA is made up of a sugar-phosphate backbone. The sugar, deoxyribose, connects to a phosphate group, and this pair, sugar and phosphate, links with others to form a long, curving chain. Attached to each sugar is one of 4 nitrogenous bases: adenine, thymine, cytosine, or guanine. These bases reach across the central axis of the helix to pair with a complementary base on the opposite strand, forming the rungs of the ladder. The pairings are consistent: adenine always with thymine, joined by 2 hydrogen bonds; guanine always with cytosine, joined by 3. This specificity is not arbitrary. It preserves the width of the helix, a uniform span of about 2 nanometers, and ensures that genetic information can be faithfully copied. The base pairs are like puzzle pieces, fitting together with a precision that allows replication and expression to unfold with stunning reliability. One strand of DNA runs in the 5 to 3 direction, while the other runs in the 3 to 5 direction. This opposing orientation is called anti-parallel. It might sound technical, but its function is simple. It allows the enzymes that work on DNA to do their jobs efficiently. In essence, it provides balance, a mirror structure that promotes harmony and function. The helical twist of DNA also creates 2 grooves along its length: the major groove and the minor groove. These grooves are not decorations. They are vital access points for proteins' molecular readers that need to interpret or replicate the DNA sequence. Imagine these grooves as quiet hallways within the spiral, where essential molecules pause to listen, to read, to act. The double helix is not a rigid ladder. It is flexible and responsive. It can bend, twist, and even form loops, allowing it to wrap neatly around histone proteins. This coiling allows meters of DNA to fit into the microscopic space of a cell nucleus. It's a kind of molecular origami, soft, efficient, and quietly ordered. When wrapped around histones, the DNA forms a structure called chromatin. Under a microscope, chromatin looks like beads on a string, each bead representing a nucleosome, a disk of histone protein, wrapped by 146 base pairs of DNA. This arrangement not only compacts the DNA, but also helps regulate access to genes. In tightly coiled regions, genes are typically silent. In loosely packed areas, genes can be read more freely. This balance of openness and closure helps control which genes are active, and when a molecular dimmer switch, adjusting the brightness of each genetic signal. During cell division, chromatin condenses further into chromosomes, distinct X-shaped structures that ensure the DNA is evenly distributed between new cells. But between divisions, the DNA relaxes back into its looser, more accessible form, ready to be read and used by the cell. The double helix is also the basis for DNA replication. When a cell prepares to divide, the 2 strands of the helix separate like a zipper, slowly unfastening, exposing the bases. Each strand then serves as a template for building a new complementary strand. Enzymes called DNA polymerases add the correct bases, one by one, following the simple rule of base pairing: A with T, C with G. The result is 2 identical double helices, each with one old strand and one new, a method called semi-conservative replication, preserving both accuracy and continuity. This structure, though microscopic, has cosmic significance. It has allowed life to exist and to evolve. It has stored the blueprints for wings and fins, bark and feathers, minds and memories. Every breath of the forest, every flick of a fish's tail, every thought you've ever had, each is shaped, in part, by the quiet spiral of DNA. It is not just a molecule. It is a memory, a record of your ancestry, a guide for your present, a gentle promise to the future. As you rest here now, breathing slowly, imagine a strand of DNA floating before you, softly glowing, turning gently in space. Picture the sugar-phosphate backbones like curved rails of light, and the base pairs like tiny bridges connecting them, one by one. See the spiral unfold, like a staircase leading upward, quietly elegant and infinitely patient. This is the shape of life's instruction, not drawn with ink or etched in stone, but sculpted in atoms in carbon, hydrogen, oxygen, nitrogen, and phosphorus. These common elements, found in stars and soil and sea, come together in a dance of exquisite precision to form the double helix, the keeper of life's quiet song. In our next part, we'll explore what these spirals contain, the genes themselves, and how they quietly govern the workings of every living being. But for now, let this image stay with you, a twisting ladder of light, carrying the soft code of life through every cell, every breath, every heartbeat. Let your thoughts drift with it, slowly, calmly, like falling leaves or clouds that barely move, and let the spiral keep turning, even as you sleep. Part 6. What are genes? Now that we've spent time with the double helix, seeing how it spirals so elegantly through the nucleus of every living cell, let us turn gently inward once more into the fine details of what it carries. For DNA, though continuous and coiled, is not an undivided stream. It is made of sections, specific, purposeful sequences. And those sequences, known as genes, are the quiet instructions behind all of life's form and function. A gene, simply put, is a segment of DNA that carries the information needed to make a specific protein or, in some cases, to guide the production of molecules that do not become proteins at all, but still perform important cellular tasks. You might think of a gene as a chapter within the long scroll of DNA, not a random portion, but a passage with a beginning, a middle, and an end, a complete thought, a sentence whispered across time. Genes are the functional units of heredity. They are the reason that traits, eye color, blood type, hair texture pass from parents to children, but their influence reaches far beyond appearance. Genes also govern the creation of enzymes that help digest food, the formation of receptors in your brain that allow you to feel emotion, and the timing of your body's internal clock. Each gene contains a code, a sequence of bases, A, T, C, and G, arranged in a particular order. This code is read in groups of 3 letters at a time. These 3-letter groups are called codons, and each codon corresponds to one of the 20 amino acids that form proteins. In this way, a gene provides a recipe, a clear, unambiguous set of steps for building a specific protein from its individual amino acid parts. Proteins themselves are the workhorses of the body. They give cells their structure. They catalyze reactions. They act as messengers, as antibodies, as transporters. And every protein begins with a gene, a short stretch of DNA written in the language of nucleotides. But not every section of DNA is a gene. In fact, only a small fraction of your genome, about 1 to 2%, consists of protein-coding genes. The rest includes regulatory sequences, non-coding RNAs, repeated segments, and vast regions whose functions we are still learning. This non-coding DNA was once called junk, but science now sees it differently. Like the spaces between words in a sentence or the quiet pauses in a piece of music, non-coding DNA plays its own subtle role in the rhythm of life. To begin the process of turning a gene into a protein, the DNA is first transcribed into messenger RNA, or mRNA. This RNA is a close copy of the gene sequence, similar in shape and language, but with a small variation. The base thymine, T, is replaced by uracil, U. This RNA strand is then processed and sent out of the nucleus to a ribosome, where it is translated into a protein. Each codon in the RNA is matched to an amino acid, and the ribosome strings them together like beads on a thread, forming a complete and functional protein molecule. This process from gene to protein is called gene expression, and it is highly regulated. Not every gene is active in every cell. Your eye cells, for example, express the genes needed to detect light, but not the ones needed to produce stomach acid. Liver cells express enzymes for detoxification, but not for muscle contraction. This selective gene expression gives each cell its identity, its role, and its response to the world around it. Some genes are always on, quietly humming in the background like a heartbeat, guiding essential processes. Others are activated only in certain situations, during growth, after injury, in response to light or heat or hunger. Cells adjust their gene expression based on both internal needs and external cues, a remarkable form of flexibility that allows life to adapt and respond with calm intelligence. Genes can also vary slightly between individuals. These differences, called alleles, account for much of the diversity we see in traits among people. Some alleles influence height, others affect how we taste certain foods, and still others may shape our resistance to disease. Most of these differences are harmless small changes in the genetic script that make us who we are. But sometimes, changes in a gene known as mutations can affect how a protein is made or how it functions. Some mutations have no noticeable effect. Others can lead to inherited conditions. Still, others may offer benefits, such as increased resistance to viruses or improved tolerance to certain diets. In this way, genes and their changes are part of a gentle dialogue between biology and the environment, a long conversation unfolding across generations. Humans have an estimated 0 to 25 genes. Some are short, made of just a few hundred bases. Others stretch across tens of thousands of base pairs. Yet all are composed of the same 4 letters, arranged in different sequences, like the pages of a long and ancient book. These genes are organized into chromosomes, long, bundled strands of DNA. Humans have 23 pairs of chromosomes, for a total of 46. One of each pair comes from the mother, the other from the father. Together, they carry the complete set of instructions needed to build and sustain a human life. In every cell of your body, every skin cell, every neuron, every blood cell, these chromosomes reside quietly in the nucleus, holding their cargo of genes. They are not read all at once. Only the necessary passages are opened, transcribed, translated. The rest remains tucked away, dormant, but not forgotten, ready to be called upon if needed. So what are genes? They are messages, instructions, memories, possibilities. They are the parts of DNA that speak most directly to the cell, guiding its behavior and shaping its destiny. And yet, they are only part of the story, a story that continues, softly and endlessly, within you. As you breathe slowly now, perhaps you can picture them, the genes, nestled like quiet islands along the vast sea of the genome, each one still, silent, until called upon. Each one a quiet whisper of life's plan, written in an alphabet of 4 letters, spoken not in voice, but in function. In the next part of our journey, we will look at how this vast length of DNA, all those genes and all those sequences, is packaged and organized into chromosomes. But for now, rest with the thought that within every cell there lies a script, a song, a guide, a gene, and within that, the stillness of 1,000 generations gently folded into you. Part 7. Chromosomes and Packaging DNA. By now, we've wandered softly through the structure of DNA and paused with gentle focus on genes, the functional sequences that tell a cell what to do, when, and how. But another question quietly arises. How does all of this information fit within a single cell? How does something so long, so vast in instruction, fold itself gently into the tiny space of a nucleus, no wider than a speck of dust? The answer rests in chromosomes, the elegant packages that house our genetic material. These are not static objects, not stiff containers. They are dynamic, responsive structures that change shape and organization depending on the needs of the cell. And they begin, like all things in genetics, with the double helix. If we were to uncoil the DNA in just one human cell, unspooling the spirals, smoothing the folds, straightening every twist, it would stretch over 2 meters in length. Yet it all fits, comfortably, within a nucleus just a few micrometers wide. This quiet miracle of folding and organization is made possible by a system of structural proteins, chief among them the histones. Histones are small, round proteins that act like spools. The DNA wraps itself around them, not randomly, but with regularity, about 146 base pairs per loop. These loops, called nucleosomes, resemble beads on a string when viewed under a microscope. The string is the DNA. The beads are the nucleosomes. This arrangement, known as chromatin, is the first level of DNA compaction. But the folding does not end there. The string of beads coils further, forming thicker fibers. These fibers, in turn, loop and fold into ever more compact shapes until the entire structure becomes a tightly wound, highly ordered chromosome. During cell division, these chromosomes become visible under a microscope, appearing as familiar X-shaped figures. But in non-dividing cells, they remain loosely organized, relaxed enough for the genes to be accessed and read. The way DNA is packaged is not just about space. It's also about control. Genes in tightly coiled regions of chromatin are generally silent, their instructions inaccessible, tucked away like books on a high shelf. Genes in looser regions are more likely to be active, their sequences open and available for transcription. This system allows the cell to regulate which genes are expressed, and when a graceful choreography of accessibility and silence. Humans have 46 chromosomes, arranged in 23 pairs. One chromosome in each pair comes from the mother, the other from the father. These pairs include 22 autosomes and one pair of sex chromosomes, XX for most females, XY for most males. Together, these chromosomes carry the full instruction set for building and maintaining a human body. Each chromosome is unique, not just in size, but in the genes it carries. Chromosome 1, the largest, contains over 20 genes. Chromosome 21, among the smallest, carries fewer than 300. Some regions are rich with genes, like busy neighborhoods filled with activity. Others are quiet long stretches of DNA with fewer instructions, perhaps devoted to regulatory functions or structural roles. At the tips of each chromosome are regions called telomeres, sequences of DNA that protect the ends from fraying or damage. Telomeres are like the plastic tips on shoelaces, keeping everything neat and intact. With each round of cell division, telomeres grow shorter, a soft countdown that limits how many times a cell can divide. This process is linked to aging, as cells gradually lose their ability to renew. In certain cells, like stem cells and cancer cells, an enzyme called telomerase helps replenish telomeres, allowing continued division. Near the center of each chromosome is a structure called the centromere, a pinched region where the two identical arms of a duplicated chromosome, called sister chromatids, are held together. During cell division, the centromere serves as the attachment point for spindle fibers, which pull the chromatids apart, so that each new cell receives an exact copy. This silent precision, this handing off of information, ensures that every cell in your body, whether in your heart or your fingertips, shares the same genetic blueprint. Despite this uniformity, not all cells read the same pages. The liver reads different genes than the retina. The skin expresses different instructions than the brain. The full library is always present, but each cell type selects its own chapter to read a quiet reflection of its purpose. The structure of chromosomes also helps protect the genome from harm. DNA is vulnerable to damage from radiation, from chemicals, even from the normal wear and tear of cellular processes. But the coiled structure of chromatin provides a layer of defense, and when breaks or errors do occur, the cell has systems in place to detect and repair the damage quietly, efficiently, without fanfare. Sometimes, changes in chromosome structure can lead to variations in health or development. A missing piece, an extra copy, a swapped segment, all can influence the way genes are expressed. Some of these changes are harmless. Others can lead to inherited conditions. In every case, they remind us that the structure of DNA, how it folds, where it lies, when it opens, is as important as the sequence itself. As you lie here now, in stillness, picture the chromosomes not as rigid rods, but as soft coils of ribbon folded with care, organized with patience, and quietly humming with the potential of life. Picture them floating in the gentle blue of the nucleus, not crowded, not chaotic, but calm, each one holding hundreds, sometimes thousands, of genes, each one passed on from parent to child, again and again, across the long arc of time. This is how life continues, not loudly, but gently, through silent duplication, through careful packaging, through the quiet stewardship of information. In the next part, we will explore how genes are turned into action, how the code becomes voice through the process of gene expression and transcription. But for now, rest with this thought, that in every cell there are chromosomes, not cold, mechanical things, but living scrolls, wrapped and folded like lullabies, carrying the memory of life, folded quietly, yet always ready to unfold. Part 8. Gene Expression and Transcription. Let us now explore how the silent instructions written in DNA become the actions of life, how stillness becomes motion, how code becomes function. This process, gentle and exact, is known as gene expression, and it unfolds in two quiet phases: transcription, followed by translation. Gene expression is the means by which the information stored in a gene, a segment of DNA, is used to produce a functional product, most often a protein. These proteins are the laborers of the cell. They build, repair, signal, and support. But before a protein can be made, the cell must first read the genetic instructions. This reading begins with transcription. Transcription, as its name suggests, is the act of copying a gene's DNA sequence into a related molecule called RNA, or ribonucleic acid. Imagine a scribe working quietly in the candlelit corners of a library, copying words from a sacred text. The original manuscript, the DNA is too precious to remove or alter, so instead, the cell creates a working copy, one that can leave the nucleus and be put to use. This transcription takes place inside the nucleus, where the DNA is stored. When the cell needs a certain protein, it identifies the corresponding gene and gently unwinds just that section of the double helix. Only one of the two DNA strands, the template strand, is used. The other remains untouched, its sequence protected. At this point, an enzyme called RNA polymerase binds to the DNA at a specific region known as the promoter. The promoter acts like a soft beacon, a quiet signpost that tells RNA polymerase where to begin. Once attached, the enzyme moves along the DNA, reading its bases A, T, C, and G, and building a complementary RNA strand. This new strand is called messenger RNA, or mRNA. In this RNA copy, the base uracil, U, is used in place of thymine. So wherever the DNA has an adenine, A, the RNA will pair it with a uracil. Cytosine still pairs with guanine, and thymine with adenine. The process continues smoothly, gently, like a ribbon unrolling. The RNA grows longer, letter by letter, until the entire gene has been copied. When the end of the gene is reached, a signal tells RNA polymerase to stop. It releases the newly formed mRNA strand, which is now ready to leave the nucleus. The DNA helix coils back into place, undisturbed, as if nothing had ever changed. The transcription is complete. Before the mRNA leaves the nucleus, it undergoes processing. Non-coding sections called introns are removed, and the remaining coding segments, exons, are joined together. A protective cap is added to one end, and a tail of repeating bases is added to the other. These additions help the RNA travel safely and be recognized by the machinery outside the nucleus. The mRNA then exits through small openings in the nuclear membrane, entering the broader world of the cytoplasm. There, it will find a ribosome, and the next chapter translation will begin. But for now, let us remain within the quiet bounds of the nucleus, watching this copying process unfold. Transcription is a precise and adaptable system. It allows the cell to make proteins only when and where they are needed. This regulation is part of what makes complex life possible. Different cells, neurons, skin cells, liver cells, all contain the same DNA, yet each expresses a unique set of genes. This selectivity allows them to carry out distinct roles, all while working from the same original script. The cell decides which genes to transcribe based on signals from both inside and outside. Temperature, nutrients, hormones, and even stress can influence gene expression. These signals interact with the DNA's packaging, the coiling and uncoiling of chromatin, opening certain regions for transcription while closing others. Like a librarian choosing which volumes to place on the reading table, the cell manages access with quiet intention. Sometimes, changes in gene expression are subtle, a slight increase or decrease in activity. Other times, they are transformative. A stem cell, for example, begins with no fixed identity. But as it receives cues, it begins to express a specific pattern of genes, transforming into a muscle cell, a blood cell, or a neuron. This is how the complexity of the body arises, not from chaos, but from coordinated expression, guided by the soft voices of genes being read. In some cases, transcription is controlled by transcription factors, proteins, that bind to the DNA near a gene and help RNA polymerase begin its work. These factors act like gentle hands, guiding the machinery to the correct spot, ensuring that the right genes are turned on at the right time. There are also enhancers and silencers, distant regions of DNA, that influence gene activity, even from afar. Like echoes in a canyon, they can either amplify a gene's expression or reduce it, depending on the needs of the cell. This layered regulation allows the genome to respond to subtle shifts, keeping the system balanced and responsive. The entire process of transcription, from the unwinding of DNA to the release of the mRNA, is both efficient and peaceful. No part of the DNA is consumed or damaged. It is read, copied, and closed again, like a beloved book opened and returned gently to its shelf. As you rest now, perhaps you can imagine this process, a quiet transcription, taking place in the stillness of your cells. The DNA unwinds softly. The RNA polymerase glides forward, building a matching strand. The message is formed, refined, and sent out, all of it happening without noise, without tension, just function, just flow. This is how the silent code becomes voice, how the structure becomes action, how the invisible words of DNA begin their journey toward shaping the living world. In the next part, we'll follow the mRNA out of the nucleus and into the cytoplasm, where the second phase translation will gently transform the genetic message into a working protein. But for now, let your thoughts dissolve into the rhythm of gene expression. Imagine your cells, calm and steady, transcribing their instructions in an endless, peaceful cycle, not with haste, not with error, but with the quiet steadiness of life itself. Part 9. Translation making proteins from RNA. In the soft, protected space of the cell's nucleus, a message has been transcribed, a slender ribbon of messenger RNA, copied from a gene, carrying with it the instructions to build something essential. And now, this message floats quietly through the nuclear envelope, passing into the open calm of the cytoplasm, the cell's gentle interior sea. Here, in the hush of cellular activity, begins the second phase of gene expression, translation. It is the process by which the RNA message is read and transformed into a protein, a folded chain of amino acids, ready to serve one of the many needs of the cell. To begin, the messenger RNA or mRNA must find a reader. In the cytoplasm, such a reader awaits, a softly curved molecular machine called the ribosome. These ribosomes, composed of both protein and a special type of RNA, called ribosomal RNA, rRNA, act like quiet interpreters, converting the coded sequence of the mRNA into a language the cell can use the language of proteins. The ribosome gently attaches itself to one end of the mRNA strand, near a starting codon, typically OG, which signals the beginning of the protein and codes for the amino acid methionine. This codon is like a capital letter at the beginning of a sentence, marking where the message truly begins. Once attached, the ribosome begins moving slowly along the mRNA, reading its sequence 3 bases at a time. Each group of 3, called a codon, corresponds to one amino acid. These codons are matched by small, precise molecules known as transfer RNAs or tRNAs. Each tRNA is shaped like a soft clover, with a 3-letter anticodon on one end, a sequence that can pair with a specific codon on the mRNA and an amino acid on the other. When the tRNA's anticodon finds its matching codon on the mRNA, it gently docks into the ribosome and adds its amino acid to the growing chain. Codon by codon, amino acid by amino acid, the ribosome moves along the mRNA like a hand sliding slowly across a page. The amino acids link together through peptide bonds, forming a long, unfolding chain, a polypeptide. With each new addition, the chain grows longer, its shape not yet defined, but its identity quietly taking form. Some proteins are short, just a few dozen amino acids. Others are vast, complex, and composed of thousands. But each begins this way, with the gentle reading of a message, the patient joining of pieces. As the chain lengthens, it begins to fold, not randomly, but purposefully, shaped by the unique chemistry of its amino acids. Some fold into spirals or pleated sheets. Others coil into globules or reach out with finger-like extensions. This folding is not just for form, it is for function. A protein's shape determines what it can do. Whether it will catalyze a reaction, carry a signal, form a structure, or move other molecules, all is guided by its final form. Eventually, the ribosome encounters a stop codon, one of 3 signals that mark the end of the message. No tRNA matches these stop codons. Instead, the ribosome pauses. The completed polypeptide chain is gently released, free now to fold completely into its final shape and to take up its role within the cell. The ribosome itself detaches from the mRNA, which may be read again, perhaps many times, to create more copies of the same protein, depending on the needs of the cell. Some proteins remain in the cytoplasm, working quietly in the spaces between organelles. Others are carried to different destinations. Some are packaged within vesicles, guided along the cell's cytoskeleton to the Golgi apparatus, where they are further folded, modified, or prepared for transport. A few are sent out of the cell entirely into the bloodstream, into tissues, into the wider world of the body. This entire process from RNA to protein is both flexible and finely tuned. Different cells express different genes, producing different proteins. Some are made only in response to signals. Others are created constantly, like the enzymes that allow you to breathe, or digest, or think. And yet, all proteins, no matter their type, begin in the same way, with a message, a ribosome, a sequence, a chain. The wonder of translation lies not just in the result, the finished protein, but in the clarity of its system. Each codon matches one, and only one, amino acid. The code is nearly universal. A mosquito, a mushroom, a whale, and a human all use the same codons, the same amino acids, the same basic rules. It is a shared language, unspoken, but understood by every cell of every living thing. This universality points to a deep unity in life, a common thread, a shared ancestry, carried in the very molecules of expression. And it is humbling to know that even now, as you rest in stillness, your cells are carrying out this process, transcribing, translating, reading and building, wordlessly, yet endlessly. As you lie here, let the image of the ribosome come to mind, not as a machine, but as a kind of artisan, quietly sliding along a strand of RNA, stringing together amino acids with delicate hands. Each tRNA brings its offering. Each codon is read, and a protein, shaped by sequence, begins to take form. This is translation, the making of meaning from code, the transformation of silent script into function and purpose. In the next part, we'll explore the roles proteins play in the body, not just as structures, but as the true instruments of life, working in harmony, in concert, in near silence. But for now, let your breath deepen, let your mind slow, and imagine a single strand of mRNA, gently gliding through the cell. Read not in haste, but in peace, becoming something useful, something alive, something softly made from a message in molecules. Part 10. The role of proteins in the body. Now that we've followed the quiet journey from DNA to RNA, and from RNA to a finished protein, let us take a soft step further into the world of what proteins do. These molecules, shaped and guided by genes, are not just the end of the genetic process. They are its purpose, the products of expression, and the instruments through which life unfolds. Proteins are not passive structures. They are active, responsive, and deeply involved in nearly every process inside a living cell. If DNA is the instruction manual, and RNA is the messenger, then proteins are the hands folding, building, repairing, moving. They are, in many ways, the substance of life. Let us begin with the most basic role: structure. Many proteins form the scaffolding that gives cells their shape and tissues their strength. One of the most well-known structural proteins is collagen, a long fibrous molecule that adds strength and elasticity to skin, tendons, and ligaments. Collagen winds through the spaces between cells, forming a soft framework that supports the architecture of the body. Another structural protein is keratin, found in hair, nails, and the outer layers of skin. It is tough, water-resistant, and tightly packed, protecting the body from external harm, like a quiet shield. These proteins, though never seen, are part of every movement, every soft touch, every thread of growth. Beyond structure, proteins also serve as enzymes, biological catalysts that speed up chemical reactions without being consumed. Enzymes are the quiet accelerators of life. Without them, the chemistry of living cells would proceed too slowly to sustain life at all. Each enzyme is highly specific, often working on just one type of molecule, called a substrate. The enzyme binds to the substrate like a lock to a key, holding it in just the right position to facilitate a reaction. Then, just as gently, it releases the product and moves on. This process happens countless times each second, in every cell, in peaceful coordination. For example, the enzyme amylase, found in saliva, begins breaking down starches in your mouth before food even reaches the stomach. In your cells, ATP synthase helps generate the energy molecule, ATP, quietly fueling your muscles, your brain, your breath. These reactions happen without noise or fanfare, but they are essential, ongoing, and beautifully choreographed. Proteins also act as transporters. In the blood, hemoglobin carries oxygen from the lungs to the tissues, its iron-rich structure binding oxygen molecules with a gentle grip. Within the cell membrane, transport proteins help shuttle ions and nutrients in and out, maintaining the delicate balance of the internal environment. They open and close like silent gates, responding to need, to signal, to change. In the immune system, proteins are defenders. Antibodies, also known as immunoglobulins, are proteins crafted to recognize foreign invaders, viruses, bacteria, or toxins. Each antibody is specific, binding only to its target. Once bound, it signals other parts of the immune system to respond, to neutralize the threat. These proteins do not act with violence, but with precision, identifying, binding, marking, and guiding. Some proteins act as messengers, carrying signals between cells and tissues. Hormones, such as insulin or growth hormone, are proteins that travel through the bloodstream, delivering messages that influence growth, metabolism, and balance. The body listens carefully to these signals, adjusting its behavior with quiet sensitivity. Proteins also play roles in movement. Inside muscle cells, long proteins called actin and myosin slide past one another, contracting and releasing, giving rise to every motion from the lifting of a finger to the beat of a heart. In cells, tiny protein filaments help guide the placement of chromosomes during cell division and help ferry materials from one part of the cell to another. Even the flutter of cilia, the tiny hair-like structures lining the respiratory tract, is powered by coordinated waves of protein activity. And within the nucleus, proteins help regulate which genes are expressed. Transcription factors, histones, and other regulatory proteins determine which stretches of DNA are accessible and which remain folded unread. These proteins shape the conversation between the genome and the environment, modulating how the instructions of life are carried out. What makes proteins so versatile is their structure. Each protein is made from a unique chain of amino acids, which folds into a specific three-dimensional shape. This shape determines what the protein can do, what it can bind, where it can fit, how it can move. A single change in shape, or in the order of amino acids, can alter or disrupt a protein's function. That is why the precise copying and translation of genetic information is so important. Even the softest shift in the sequence can ripple outward, affecting the body in unexpected ways. Despite their complexity, proteins are made from only 20 types of amino acids. These building blocks, arranged in various sequences, give rise to all the proteins in your body, tens of thousands of different kinds, each with its own shape, its own role, its own quiet contribution. And so, the proteins within you are not mere molecules. They are active, purposeful forms. They give your cells their identity. They allow your thoughts to form, your heart to beat, your wounds to heal. They allow you to sense the world, to adapt to change, and to carry on the quiet work of life. As you rest now, you may imagine them not as rigid machines, but as gentle forms, flowing through your bloodstream, resting within your tissues, shaping the unseen workings of the body. Picture a hemoglobin molecule, gliding calmly along, releasing oxygen into a cell. Picture an enzyme, folding a molecule into something new. Picture a signal protein moving across a membrane, carrying word of what is needed, what is changing. These are the quiet heroes of your biology. They ask for no recognition. They make no sound. But without them, there would be no form, no function, no self. In the next part of our journey, we'll explore how genes passed from one generation to the next carry variations that create the subtle uniqueness in every person. But for now, rest in the knowledge that within you, proteins are at work calm, tireless, and always in harmony with life. Soft structures, performing quiet miracles. Part 11. Genetic variation and inheritance. As we continue our peaceful journey through the world of genetics, let us now turn to a question as ancient as life itself: how traits are passed from one generation to the next, and why no two living beings are exactly alike. At the center of this gentle mystery lies genetic variation, the quiet differences in DNA that give rise to the rich diversity of life, and with it, the concept of inheritance, through which these differences, these soft patterns, are carried forward not loudly, but subtly, from parent to child, from ancestor to descendant. Each human being has two complete sets of chromosomes, one set of 23 from their mother, and another 23 from their father, for a total of 46. This pairing is the basis of inheritance. For every gene in your body, you carry two copies, one maternal, one paternal. These copies may be identical, or they may differ. These different versions of a gene are called alleles. Some alleles influence traits that are easily seen: the curl of hair, the shape of an earlobe, the ability to roll one's tongue. Others govern hidden processes: the rate of metabolism, the expression of hormones, the timing of growth. Most traits are shaped not by one gene, but by many, their effects layering together softly, shaping us with quiet complexity. The rules that describe how alleles are passed on were first uncovered by Gregor Mendel, the monk and gardener, whose careful study of pea plants laid the foundation for modern genetics. He observed that certain traits followed consistent patterns of inheritance, dominant traits appearing even when only one copy of the allele was present, while recessive traits appeared only when both copies matched. Imagine a gene with two alleles, one for brown eyes, another for blue. If brown is dominant, a person with one brown and one blue allele will have brown eyes. Only those with two blue alleles will have blue eyes. These patterns, known as Mendelian inheritance, are simple in form, yet foundational. But many traits are more complex. They may be polygenic, influenced by multiple genes, as with height, skin tone, or intelligence. Others are influenced by both genes and the environment, a symphony of nature and nurture playing together. The source of genetic variation is rooted in three quiet mechanisms: mutation, recombination, and independent assortment. A mutation is a change in the DNA sequence. It may be as small as a single swapped base, or as large as a missing section of a chromosome. Most mutations occur naturally during DNA replication or under the influence of environmental factors. Some are neutral, some beneficial, and some harmful, but all serve as the raw material for evolution, a slow, quiet source of change and possibility. Recombination occurs during the formation of reproductive cells, eggs, and sperm. As chromosomes are prepared for passage to the next generation, Sections of maternal and paternal DNA are exchanged. This gentle shuffling creates new combinations of alleles, blending the traits of grandparents, great-grandparents, and generations beyond. Independent assortment adds another layer. When eggs and sperm are formed, each pair of chromosomes is sorted independently of the others. This means that the inheritance of one trait does not affect the inheritance of another. The result is an astonishing number of possible combinations, a quiet explosion of uniqueness in every generation. Together, these processes ensure that no individual is exactly like another, not even identical twins, who may still show small differences due to environmental influences and epigenetic changes. Variation is everywhere, and it is natural. It is why a family may share a resemblance, yet each member remains distinct. It is why populations adapt over time, as new combinations of traits rise and fall in gentle response to changing conditions. Genetic variation also underlies the study of hereditary conditions. Some conditions, like cystic fibrosis or sickle cell anemia, are linked to specific gene mutations. Others, like heart disease or diabetes, arise from the interaction of many genes with lifestyle and environment. Understanding these links allows medicine to move quietly toward prevention, treatment, and healing. Beyond the body, variation allows scientists to study ancestry, tracing the silent trails of migration, connection, and kinship through strands of DNA. It is now possible to peer into the past through genetic markers, to see echoes of ancient populations, to understand how people spread across the earth, shaped not by conquest, but by continuity. Inheritance also plays a key role in agriculture, where the selection of traits has shaped crops and animals for millennia. Farmers, long before the word gene was known, selected plants for sweetness, resilience, or color. Over time, these chosen traits became part of the genetic fabric of domesticated species, a soft record of human care and of nature's quiet response. There is, too, a kind of poetry in inheritance. The curve of a smile passed down through generations, the way a grandparent's manner appears unexpectedly in a child. Traits carried not just in behavior, but in biology. Each strand of DNA is a braid, part mother, part father, and part ancient lineage, reaching far beyond memory. And so, while genes shape us, they do not define us completely. They provide the template, the possibility. But the outcome, who we become, is shaped also by experience, by chance, by love, by learning. The genome offers the notes, but the melody is played in real time, in real lives. As you rest now, you might reflect on your own inheritance, not just the traits you've been given, but the gentle legacy of those who came before. Their genes live in you, not as a burden, but as a foundation, a whisper of the past, carried into the present. And with each breath, with each moment of stillness, you continue that story, shaping it quietly, uniquely, into your own. In the next part, we'll explore how mutations, these small, spontaneous changes in the genetic code, arise and influence life. But for now, let this moment settle. Let the idea of inheritance rest within you, like the echo of a lullaby passed down across generations. Not loud, not rigid, but soft, enduring, and beautifully human. Part 12. Mutations, Natural Changes in DNA. As we continue our quiet journey through the world of DNA, we arrive now at a gentle turn in the path. The idea that even this elegant molecule, for all its order and structure, is not fixed. It changes. It adapts. It evolves. And the name we give to these changes, when they happen within the sequence of DNA, is mutation. In everyday language, the word mutation may sound sudden or alarming, but in biology, it is neither. It is natural, frequent, and often subtle. Mutations are the soft, silent shifts in the letters of the genetic code, the quiet variations that occur as cells copy their DNA, as they divide, as they live. Most go unnoticed. Some have consequences, but all are part of the gentle motion of life. At its simplest, a mutation is a change in the sequence of bases, the A's, T's, C's, and G's that make up DNA. These bases form the words and phrases of the genetic code. And just as a single letter change in a sentence can alter its meaning, so too can a base change affect the function of a gene. But not always. Much of DNA can tolerate change without harm. Some areas are flexible. Others are deeply conserved, protected by evolution because their sequence is essential. There are several types of mutations. The most common is the point mutation, where a single base is swapped for another. This is like replacing one letter in a word. Sometimes the meaning stays the same. Sometimes it changes. If the altered codon still produces the same amino acid, the mutation is called silent. If it changes the amino acid, it is a missense mutation. And if it creates a stop codon too early, it is a nonsense mutation, cutting the protein short. There are also insertions and deletions, the quiet addition or removal of bases. These can shift the reading frame of a gene, altering every codon that follows. Such frame shift mutations can be disruptive, but they can also, at times, lead to new forms, new possibilities. Mutations can occur spontaneously as a result of natural errors in DNA replication. Every time a cell copies its genome, all 3 billion base pairs, it relies on enzymes that work with remarkable accuracy. But even these enzymes, with their proofreading and correction mechanisms, make mistakes now and then, perhaps once in every 100 million bases. Over time, these rare slips accumulate, gently adding variation to the genome. Mutations can also be induced by environmental factors, certain chemicals, ultraviolet light, or radiation. These influences may damage the DNA, leading to breaks or mispairings. The cell works tirelessly to repair such damage. Specialized enzymes scan the DNA for irregularities, remove faulty sections, and rebuild the correct sequence. But some damage escapes notice, and these uncorrected changes become part of the genetic record. Not all mutations occur in genes. Many happen in non-coding regions, stretches of DNA that do not produce proteins. Some of these regions help regulate gene activity, so mutations there can still influence expression. Others may have little to no immediate effect. Over long periods of time, such changes can accumulate, marking the passage of generations like soft pencil notes in the margins of a book. Some mutations are inherited, passed down from parent to child. These are called germline mutations because they originate in the egg or sperm. Others arise later, in the body's somatic cells, and are not passed on. These somatic mutations can influence health, especially if they occur in genes that control cell division, potentially leading to cancer, a condition in which cells grow without limit, guided by disrupted instructions. And yet, while some mutations may be harmful, many are neutral, and a few may even be beneficial, conferring a resistance to disease, a tolerance to environment, or an adaptation to a new challenge. These helpful mutations, though rare, are the seeds of evolution, passed on through generations. They may become common in a population, shaping the traits of species over time. This is the quiet power of mutation, not chaos, not error, but variation, the gentle shifting of life's possibilities. It is how flowers develop new colors, how birds adapt to new climates, how humans have grown to digest new foods, resist new viruses, and thrive in environments from polar cold to desert heat. Mutation also explains why no genetic blueprint is perfectly identical from one person to another. Even identical twins, who share the same original DNA, may differ by a few somatic mutations or by how their genes are regulated. Each person, in this way, is a variation, a soft experiment in form, function, and identity. Modern science has found ways to read these mutations, to compare genomes, to track changes across populations and time. These efforts, unfolding quietly in laboratories and research centers, help us understand disease, trace ancestry, and glimpse the long arcs of evolution. But at its heart, mutation remains a simple idea that the code of life is not written in stone, it is written in molecules. And molecules move, they shift, adapt, evolve. As you rest here now, you might picture the genome not as a fixed blueprint, but as a living document, one that rewrites itself slowly, subtly, with each generation. Picture a single base, changing from a C to a T, a single letter in a 3 billion letter book. It happens without sound, without alarm, and yet it becomes part of the story, part of you. In the next part of our journey, we'll look more closely at how the entire DNA strand is faithfully copied during cell division, the elegant process of replication. But for now, let this thought rest gently in your mind, that within you, within all living things, the code of life is flexible. It listens, it shifts, it changes, quietly and endlessly. Part 13. DNA replication. As we continue our gentle journey through the processes of life, we come now to one of biology's most elegant and vital acts, the quiet copying of DNA. Known as replication, this process ensures that every cell, before it divides, carries with it an exact copy of its genetic information. It is not a loud or hurried event. It happens deep within the nucleus, often while the world outside remains unaware. And yet, with each replication, the continuity of life is quietly preserved. DNA replication begins at special locations on the double helix known as origins of replication. These are specific sequences that act like quiet doorways, signaling where the copying should begin. In a human cell, there are thousands of these origins, allowing the long strands of DNA to be duplicated swiftly and efficiently. When replication begins, enzymes, called helicases, gently unwind the two strands of the DNA double helix. Like parting the threads of a delicate braid, the hydrogen bonds between the base pairs are unzipped, leaving each strand exposed open, not damaged, ready to serve as a template for its mirror image. These single strands, however, are not left unsupported. Single-strand binding proteins attach to the separated strands, keeping them apart and stable. Meanwhile, another enzyme called topoisomerase works just ahead of the unwinding fork, relieving any twisting tension in the DNA, preventing knots or tangles from forming. Now the two single strands are ready to be copied. And this is where the enzyme DNA polymerase begins its work. DNA polymerase moves along each strand, reading its sequence, and building a new complementary strand one base at a time. Where there is an A, it adds a T. Where there is a C, it adds a G. The pairing rules are simple, but the process is astonishing in its precision. Replication happens in opposite directions on the two strands. Because the strands of DNA run in opposite orientations, one from the 5 to 3 direction, and the other from 3 to 5. DNA polymerase can only add new nucleotides in the 5 to 3 direction. Because of this, replication is smooth and continuous on one strand called the leading strand, and more intricate on the other, known as the lagging strand. On the lagging strand, DNA is copied in short segments called Okazaki fragments, each begun with a small RNA primer laid down by the enzyme primase. These fragments are later joined together by another enzyme, DNA ligase, which acts like a soft stitcher, sealing the nicks, forming a continuous strand. Throughout this process, The cell employs layers of proofreading. DNA polymerase checks its work, pausing at any mismatch, removing the incorrect base, and replacing it. Additional repair enzymes scan the new DNA for any remaining errors, gently correcting them with care. Because of these systems, DNA replication is remarkably accurate, with only about one error in every 10 billion bases copied. The entire genome, all 3 billion base pairs in humans, is duplicated before each cell divides. And once the copying is complete, the cell has 2 full, identical sets of DNA. These are packaged into chromosomes, each now consisting of 2 identical sister chromatids, joined at a central point called the centromere. When the time comes for the cell to divide through a process called mitosis, these chromatids will be separated, with one copy going to each new daughter cell. In this way, the genetic information is passed on, not diminished, not altered, but preserved. This act of replication is the basis for growth, healing, renewal. Each time your skin replaces its cells, each time your body recovers from a small wound, each time a child grows taller, DNA replication has quietly made it possible. It is a process of continuity, not dramatic, not visible, but deeply essential. It allows life to echo forward, cell by cell, moment by moment, always carrying the same instructions, faithfully read and carefully duplicated. In its earliest form, replication likely evolved in the very first living cells, billions of years ago. The ability to copy genetic material, to pass it forward, marked the beginning of biological inheritance, the soft pulse that connects all living things, from bacteria to blue whales, from ancient ferns to your own fingertips. Today, in every one of your cells, replication is still happening, cells preparing to divide, to grow, to replace what has been lost. And in each replication, the same care is taken, the same enzymes, the same rules, the same silent choreography. As you lie here now, perhaps you can imagine a single strand of DNA slowly unwinding. Picture the helicase opening the helix, the polymerase gliding along, matching letters, building a partner, the new strands curling gently into place, forming once again the timeless double helix, complete, accurate, alive. This is replication, the self-copying of life, a quiet assurance that what has come before will continue not by chance, but by careful molecular memory. In the next part of our journey, we'll turn to Mendelian genetics, the patterns of inheritance first discovered in a monastery garden, long before DNA was known. But for now, let this image remain the steady unwinding of DNA, the faithful building of new strands, the quiet preservation of life's code. Nothing rushed, nothing wasted, only the soft continuity of what has always been passed down, strand by strand, breath by breath. Part 14. Mendelian genetics made simple. Long before the discovery of DNA, before microscopes revealed the inner world of the cell, and before the twisting double helix was known to science, there was a garden quiet, green, and full of pea plants. And in that garden, in the 19th century, a patient monk named Gregor Mendel conducted a series of gentle experiments that would softly lay the foundation for all of modern genetics. Mendel worked not with advanced tools, but with nature itself. He noticed that when he crossed certain varieties of pea plants, some tall, others short, some with smooth seeds, others with wrinkled ones, the traits did not blend or scatter at random. Instead, they followed clear, consistent patterns. These observations, repeated and recorded with quiet care, revealed how traits are passed from one generation to the next. Today, this field of study is known as Mendelian genetics, and though simple in principle, it remains a vital part of how we understand inheritance. Let us begin with Mendel's most fundamental idea, that traits are determined by discrete units, now known as genes. Each gene has different versions called alleles, and these alleles can influence how a trait appears. For each gene, an organism inherits two alleles, one from each parent. These alleles may be the same, or they may differ. If both alleles are the same, the organism is said to be homozygous for that gene. If the alleles are different, it is heterozygous. Some alleles are dominant, meaning they will determine the trait even if only one copy is present. Others are recessive, meaning their influence is hidden unless both alleles are the recessive form. Consider a simple example: a gene that influences flower color in pea plants. Suppose the dominant allele, which we can call P, produces purple flowers, while the recessive allele, P, produces white flowers. A plant with the genotype PP will have purple flowers, both alleles are dominant. A plant with POM will also have purple flowers, the one dominant allele is enough. Only a plant with PP will have white flowers, both alleles are recessive, and so the trait is expressed. These patterns are consistent and predictable. Mendel summarized them in two quiet principles. The first is the law of segregation. This law states that each parent carries two alleles for each gene, but passes only one to each offspring. This separation of alleles occurs during the formation of reproductive cells, eggs, and sperm, ensuring that every child receives one allele from each parent. The second principle is the law of independent assortment, which states that alleles for different genes are passed independently of one another. The inheritance of flower color, for example, does not influence the inheritance of seed shape. This is because genes are often located on different chromosomes and are sorted separately during the formation of gametes. To visualize these patterns, scientists often use a simple diagram known as a Punnett square, a quiet little grid that shows all the possible combinations of alleles from two parents. It's a gentle tool, not complex, but useful for predicting the likelihood of different traits appearing in offspring. Imagine two pea plants, both heterozygous for flower color, PP. In a Punnett square, we place one parent's alleles across the top and the other parent's down the side. Each box inside shows one possible genetic outcome. One box shows PP purple flowers. Two boxes show PP also purple. One box shows PP white flowers. This gives a ratio of 3 purple to 1 white, or 3:1, a classic Mendelian pattern. Mendel's work extended beyond single genes. He studied multiple traits, including seed color, pod shape, and plant height. In every case, he observed that the patterns of inheritance were orderly and mathematical. Traits didn't merge or fade. They were passed on, like quiet notes in a song, distinct and deliberate. Of course, not all traits follow these simple Mendelian rules. Many are influenced by multiple genes, by environmental factors, or by more complex patterns of dominance and interaction. But the principles Mendel discovered still serve as the gentle backbone of genetic science, a foundation upon which deeper understanding has grown. Mendel's work was not recognized in his lifetime. It wasn't until decades later, at the dawn of the 20th century, that scientists rediscovered his papers and realized their significance. By then, biology had begun to glimpse the cell, the chromosome, and soon the molecule of DNA itself. And within those discoveries, Mendel's quiet patterns found their molecular basis. We now know that alleles correspond to slightly different versions of a gene's DNA sequence. Dominant alleles often code for functional proteins, while recessive ones may result from changes that disrupt the gene's expression. Yet the basic idea remains unchanged, that traits are guided by the pairing of alleles, passed gently from parent to child. Mendelian genetics also helps us understand certain inherited conditions. For example, cystic fibrosis is caused by a recessive allele. A person must inherit 2 copies, one from each parent to develop the condition. Carriers, who have only one copy, do not show symptoms, but can pass the allele to their children. The same principle applies to many other conditions: sickle cell anemia, Tay-Sachs disease, phenylketonuria. By understanding inheritance, families and doctors can anticipate risk, offer guidance, and provide care all through knowledge rooted in a simple garden, a patient observer, and the patterns of pea plants. As you rest now, perhaps you can picture Mendel in his quiet greenhouse, gently tending his plants, watching, noting, and asking the simplest of questions. How do traits pass from one generation to the next? And with each blossom, each seed pod, each carefully counted trait, he listened, and slowly the answers emerged. Not with noise, not with urgency, but with gentle clarity. In the next part of our journey, we will explore the quiet world beyond the genes into epigenetics, where the expression of genes can be softened or strengthened by signals and structures outside the DNA sequence itself. But for now, let the soft simplicity of inheritance rest with you. Pairs of alleles, patterns unfolding, life continuing patiently, predictably, and peacefully. Part 15. Epigenetics, more than just DNA. Until now, we've gently followed the story of DNA as a code, a long, silent sequence of letters passed down from generation to generation. We've seen how genes shape proteins, and how inheritance quietly directs the flow of traits. But what if the story doesn't end with the sequence? What if there are softer, more subtle ways to influence the activity of genes without changing the code itself? This is the quiet world of epigenetics, a realm where genes are still, but can be awakened, where some are gently silenced, others softly enhanced, where the cell doesn't rewrite the script, but adjusts how it is read. The word epigenetics comes from the Greek prefix epi, meaning upon or above. It refers to the layers of regulation that rest atop the DNA sequence modifications that influence gene expression without altering the underlying genetic code. These changes do not rewrite the letters of DNA, but they can determine which pages are opened and which are kept closed. Imagine DNA as a long scroll, carefully stored in a library. Every gene is a paragraph, a sentence, a message. Epigenetics is like placing bookmarks, adding soft highlights, or folding corners of pages. The words remain the same, but the experience of reading changes. One of the most studied epigenetic mechanisms is DNA methylation. In this process, a small chemical group, a methyl group, is attached to certain bases in the DNA, most often at cytosine. When a gene's promoter region is heavily methylated, the gene is typically silenced, its message unread. Methylation acts like a gentle, do not disturb sign. It doesn't erase the gene, but makes it less accessible. This process is essential for normal development, for turning off genes when they're no longer needed, and for maintaining cellular identity. Another important layer is histone modification. Recall that DNA is wrapped around proteins called histones, forming a structure known as chromatin. These histones are not passive supports, they are responsive. Small chemical tags, such as acetyl groups or phosphate groups, can be added to the histone tails, influencing how tightly or loosely the DNA is wrapped. When the wrapping is loose, genes are more likely to be expressed, their sequences open and available. When tight, the DNA becomes hidden, silent. These histone modifications act like dimmer switches, adjusting the brightness of gene expression in a smooth, responsive way. Epigenetic changes are not random. They can be influenced by signals from within the body and from the outside world, by hormones, nutrients, stress, and even temperature. The foods we eat, the air we breathe, the thoughts we carry all may leave small chemical footprints on the genome, shaping how it speaks. In early development, epigenetic marks guide cells as they specialize. A skin cell, a neuron, a muscle fiber, all arise from the same genome, yet express different genes. These differences are maintained not by changes in the DNA sequence, but by layers of epigenetic information that tell each cell which parts of the script to read. Epigenetics also plays a role in imprinting, a process where certain genes are expressed only from the allele inherited from one parent, either the mother or the father. The other copy is epigenetically silenced. This phenomenon is rare, but deeply significant, influencing growth, development, and metabolism. Perhaps most fascinating is that some epigenetic changes can be passed on from one generation to the next. Though most are reset during the formation of sperm and egg, a few marks may remain quiet echoes of a parent's environment or experience carried forward into their children. This has led scientists to explore the connections between epigenetics and long-term health. Can early nutrition affect not only the individual but their descendants? Can stress, experienced before birth, shape gene expression across generations? These are questions not yet fully answered, but they speak to the depth of epigenetic influence, its power to reach beyond time. In some cases, disruptions in epigenetic regulation can lead to disease. If a tumor suppressor gene is silenced by methylation, for example, a cell may begin to grow uncontrollably. Researchers are now exploring therapies that can gently reverse such marks, reactivating silenced genes, restoring balance without altering the genetic code. And so, the genome is not a static document. It is dynamic, sensitive, and responsive. Epigenetics is the system through which the body listens, adapts, and adjusts. It is the gentle hand that guides which genes are voiced and which remain quiet. As you lie still now, perhaps you can imagine your DNA wrapped gently around histones, marked with tiny chemical tags, methyl groups resting on cytosines, acetyl groups softening histone tails, a landscape of information not loud but quietly instructive. Each cell in your body carries not only the same DNA, but its own unique pattern of epigenetic marks. This is what gives a liver cell its shape and function, even though its DNA is identical to that of a neuron or a muscle fiber. This is the soft artistry of epigenetics shaping identity, not by changing what is written, but by choosing how it is read. And just as your body changes, so too can these marks. Some may fade with age, others shift with the seasons of life. All the while, your genome listens and responds gently, patiently, without noise. In the next part of our journey, we will explore how DNA and genes are used in modern science, in medicine, ancestry, and research. But for now, rest with this thought, that the story of life is not told by sequence alone. It is also shaped by silence, by folding, by soft, chemical whispers. And in every quiet cell, the story continues. Part 16. DNA in modern science. Through the quiet threads of DNA and the gentle mechanisms that shape its expression, we've come to understand life not just as a collection of traits, but as a soft and flowing code, one that adapts, responds, and continues. And now, in this modern age, we find ourselves in a time when that code can be read, explored, and even softly guided. In the last few decades, DNA has become more than a molecule of inheritance. It has become a key unlocking mysteries in medicine, ancestry, agriculture, and beyond. Scientists now work with DNA as both map and memory, treating it not as something distant, but as a quiet, living companion to human understanding. One of the most significant milestones in modern science was the Human Genome Project, completed in the early 2000s. This global endeavor sought to read base by base the entire sequence of human DNA. It took more than a decade, involved hundreds of researchers, and revealed over 3 billion base pairs, forming the full blueprint of our species. From this project came not just information, but perspective. We learned that human beings share over 99.9% of their DNA with each other, that the remaining fraction, less than 1%, accounts for the subtle differences in appearance, metabolism, susceptibility to disease, and more. It was a reminder, both humbling and profound, of how deeply we are connected. Today, reading DNA through genetic sequencing is faster and more accessible than ever. What once took years can now be done in hours. Through small samples of saliva or blood, a person's genome or parts of it can be analyzed to look for markers, traits, or variations. In medicine, this has opened the door to personalized healthcare. By understanding a patient's genetic makeup, doctors can predict how they might respond to certain medications or which conditions they might be more vulnerable to. This approach, called pharmacogenomics, allows treatments to be matched not just to symptoms, but to the patient's unique biology. Genetic testing is also used to identify inherited conditions. Mutations in specific genes can signal an increased risk for diseases such as breast cancer, colon cancer, or cardiomyopathy. Early awareness can guide decisions not through fear, but through quiet preparation and care. Beyond disease, DNA is also used to trace ancestry. Companies now offer genetic ancestry testing, showing people where their forebears may have lived and how their DNA connects them to ancient populations. These tests compare your genetic markers to large databases, revealing patterns that stretch across continents and centuries. For many, this brings a sense of identity, a soft bridge between the present and the past. In forensics, DNA plays a quiet but crucial role. A single strand left behind on a piece of cloth or a strand of hair can help identify individuals, solve crimes, or reunite families. Because DNA is unique to each person, except in identical twins, it acts like a biological fingerprint, silent, yet unmistakable. In agriculture, DNA is used to improve crops, making them more resistant to drought, pests, or disease. By understanding the genes that control traits such as yield, taste, or nutrition, scientists can guide the growth of plants with greater precision, helping to feed a growing world while preserving natural balance. In conservation biology, DNA helps track endangered species, understand populations, and preserve genetic diversity. By studying the DNA of animals and plants, scientists can make informed choices about breeding, habitat protection, and species recovery, working not just to save life, but to protect its quiet complexity. Modern science has also developed tools to edit DNA. One of the most talked about methods is called CRISPR-Cas9, a system borrowed from bacteria that allows scientists to cut and modify DNA at precise locations. This technique holds promise for treating genetic disorders, correcting mutations, and even preventing certain diseases before they manifest. Yet even here, the approach is soft and deliberate. The aim is not to rewrite the story of life, but to better understand it, to guide it gently, responsibly, and with care. It's important to note that while these technologies offer great promise, they also raise questions: ethical, philosophical, and practical. As we learn to read and shape the code of life, we must do so with humility, remembering that DNA is not just a tool. It is a reflection of life's deep history of processes shaped not by force but by time, adaptation, and balance. Scientists now also study the microbiome, the DNA of the trillions of bacteria that live in and on our bodies. These tiny organisms, each with their own genomes, influence digestion, immunity, and even mood. Understanding their DNA has opened new paths in medicine, nutrition, and mental health, reminding us that the story of genetics is not just human, but shared. As you rest now, perhaps you can imagine a scientist at work not in a loud lab of clanging machines, but in a quiet room filled with soft light and steady thought, reading sequences, comparing patterns, asking questions, searching, gently, for understanding. Perhaps you can imagine a strand of DNA, unfolding under the lens of discovery, not dissected but honored, its bases shining like faint stars, their order revealing a truth both specific and shared. Modern science has given us the tools to explore DNA with clarity and care, but the wonder remains not in what we can do, but in what DNA is: a silent, living language, a bridge from cell to cell, from generation to generation, from species to species. In our final part, we will reflect on this journey, the quiet unity of life, the shared code that connects us all. But for now, let the thought of DNA in modern science settle into you like a gentle breeze, guiding, not pushing, discovering, not disrupting. A molecule once unseen, now understood, still quiet, still sacred. Part 17. Closing reflections on our genetic blueprint. And now, as we reach the final stretch of our quiet journey through the world of DNA and genes, we pause not with urgency, but with gentle reflection. For we have wandered softly through the spiral staircase of the double helix. We have traced the movement of genetic instructions from molecule to protein and followed the echo of inheritance from parent to child. We have explored the subtle shifts of mutation, the careful copying of replication, and the dimming and brightening of gene expression through epigenetic whispers. We have seen DNA not just as a biological structure, but as a calm, enduring language. It is a language without sound, a script without punctuation, a message passed down in silence, strand by strand, cell by cell, life by life. Every one of your cells, save a few red blood cells and fragments like platelets, contains this message. It is tucked quietly inside the nucleus, coiled with care, but always ready to be read. This code does not change with the wind or mood. It persists, soft and consistent, through your growth, your aging, your healing. And yet, it is not fixed like stone. It bends with experience. It listens to environment. It adapts through regulation and repair. The DNA within you is not simply a set of instructions. It is a living record, a biological memory of everything that has come before, from the first spark of life in ancient oceans to your mother's voice, your father's eyes, your first breath. Consider the deep connection that this blueprint provides. The same four letters A, T, C, and G build not only you, but also whales and butterflies, sunflowers and moss, elephants and eagles. These letters, repeating in varied rhythms, are the common thread that runs through all life. The same base pairs, the same molecular bonds, the same quiet laws. You and a tree, different as you may seem, share this code. You and the stars, whose atoms helped form the sugars and phosphates of your DNA, are linked by origin. Life, in its most essential structure, is a single story told in the language of molecules, shaped by time and always evolving. And yet, for all this shared code, each human being is unique. The specific sequence of your genome, the precise arrangement of bases that defines your blueprint, exists nowhere else in exactly the same way. Not in anyone alive today. Not in anyone who has ever lived. You are one variation in the great, ongoing song of life, a softly distinct note in a symphony that stretches back billions of years. And this uniqueness, this quiet singularity, comes not just from your DNA, but from the way it unfolds in you, the genes that are turned on or off, the environments that shape you, the thoughts, feelings, and choices that no genome can predict. So while DNA provides the foundation, you are the living expression of that foundation. More than a code, more than a sequence. You are a moment of biology, becoming being. It is a comfort, perhaps, to know that this blueprint, this coiled, whispering scroll, is not something you must control. It does not need your awareness to work. Your cells read it on their own. Your proteins fold without instruction. Your body heals, grows, adapts, guided by a code that does not ask to be seen. And in sleep, when the conscious mind drifts away, this blueprint continues its work. Cells divide, genes are expressed, enzymes carry out their tasks, all while you rest in quiet stillness. As you lie here now, at the edge of waking and dreaming, imagine this blueprint resting within you, not glowing, not flickering, but steady. Picture the double helix, spiraling gently. Picture the chromosomes, coiled and content. Picture a single gene, its bases paired like tiny, silent bridges, waiting patiently to be read. Let yourself feel the calm that this order brings. A system so old, so tested, so quiet, and yet so dependable. It does not rush. It does not waver. It simply continues. In each breath, each heartbeat, each peaceful moment. You are the result of this quiet choreography, the product of molecules and time, of inheritance and expression. You are not separate from nature. You are its continuation, a soft branch on the tree of life, a single voice in the endless biological hymn. As this journey comes to rest, let that thought remain with you. You are made of DNA, the same molecules that built your ancestors, that shaped your features, that guided your first cells into form. This code does not define your worth, but it holds your history. It does not dictate your path, but it gives you the tools to grow, to change, to be. And so, you may sleep now with a deeper stillness, knowing that within you lies an archive, a blueprint, a soft and faithful pattern. It will be there when you wake. It will be there as you move through the world, and it will always speak, quietly, in the language of life. Rest now, and let the blueprint carry on.